May 14, 1943. Deep within a converted ice cream factory in Terre Haute, Indiana, Dr. Margaret Hutchinson Rousseau stood before a fermentation tank the size of a small automobile, watching golden liquid drain into collection vessels below. The substance flowing through those tubes represented something pharmaceutical executives had declared impossible. Penicillin, the miracle drug that could cure infections no other medicine could touch, produced not in laboratory quantities measured in milligrams, but in industrial volumes measured in gallons. Every expert in America had told her it couldn't be done. Fleming's original mold grew in flat glass dishes, producing barely enough penicillin to treat one patient after weeks of cultivation. The drug's chemical structure was so unstable that it degraded within hours of extraction. Pharmaceutical companies had invested millions trying to scale production, only to achieve yields so microscopic that treating a single the soldier required the entire output of half a dozen laboratories working for weeks. But Rousseau had ignored them all. And on this spring morning, in a Midwest factory that still smelled faintly of vanilla, her team was about to prove that the impossible had become routine. If you're enjoying this deep dive into the story, hit the subscribe button and let us know in the comments from where in the world you are watching from today. The penicillin crisis of 1942 existed at the intersection of medical miracle and industrial catastrophe. Alexander Fleming had discovered the antibacterial properties of Penicillium notatum mold in 1928 at St. Mary's Hospital in London, observing that a contaminated petri dish had killed surrounding Staphylococcus bacteria. His published findings described a substance of remarkable potency, capable of destroying infectious organisms while leaving human tissue unharmed. For a decade, the discovery remained a laboratory curiosity, too difficult to produce in meaningful quantities for clinical use. The breakthrough that transformed penicillin from scientific oddity to medical necessity occurred in 1940 at Oxford University, where Howard Florey and Ernst Boris Chain led a team investigating antibacterial agents. Their research demonstrated that purified penicillin could cure previously fatal bacterial infections in mice with efficiency that exceeded any known treatment. Human trials conducted in early 1941 showed similarly dramatic results. Patients dying from septicemia recovered within days after receiving injections of the drug. Wounds that would have required amputation healed cleanly when penicillin prevented bacterial infection. But the quantities required to demonstrate these miraculous recoveries had nearly bankrupted Oxford's research budget. Treating a single patient required penicillin extracted from 2,000 litres of mold culture broth, a process that took 6 weeks, and required equipment occupying an entire laboratory. Florey's team had resorted to collecting patients' urine to recover excreted penicillin for reuse, a desperate measure that illustrated how far the supply situation had degraded from any practical medical application. The mathematics of wartime demand made Oxford's production methods laughably inadequate. British military medical officers estimated that treating battlefield casualties from a single major engagement would require more penicillin than all of Britain's laboratories could produce in a year. Infected wounds, the leading cause of combat deaths after immediate battlefield trauma, killed thousands of soldiers who survived their initial injuries. Pneumonia, meningitis, and septicemia devastated military hospitals, where doctors could only watch as infections consumed patients whose wounds had been survivable. American pharmaceutical companies received the penicillin challenge in summer 1941 when Florey traveled to the United States seeking industrial partners capable of scaling production beyond laboratory methods. His meetings with executives at Pfizer, Merck, and Squibb revealed immediate problems that threatened to prevent penicillin from ever reaching mass production. The drug's instability meant it degraded rapidly at room temperature, making storage and transport nearly impossible. The Penicillium mold required specific growing conditions that were difficult to maintain in industrial settings. Most critically, yields from deep tank fermentation, the only method capable of producing commercial quantities, were so low that pharmaceutical executives questioned whether industrial production would ever be economically viable. Pfizer's initial experiments with deep tank fermentation had produced results that confirmed industry skepticism. Growing penicillium in large fermentation vessels, the same equipment used for producing citric acid and other industrial chemicals, should have scaled production proportionally to tank volume. Instead, yields dropped precipitously as tank size increased. A 1,000-gallon fermentation tank produced less penicillin per gallon of culture medium than Fleming's original Petri dishes, a result that defied every principle of industrial chemistry. The technical obstacles stemmed from fundamental characteristics of penicillium metabolism that were poorly understood in 1941. The mold required constant oxygen supply throughout the fermentation process, but oxygen transfer into deep liquid cultures proved extremely difficult at industrial scales. Temperature control was critical. Variations of just 2 degrees Fahrenheit could kill entire batches. The culture medium required precise mixtures of corn, steep liquor, lactose, and mineral salts, with ratios that changed as the fermentation progressed. Any contamination by competing bacteria or fungi would destroy the batch, and maintaining sterility in 1,000-gallon tanks presented challenges that exceeded existing industrial capabilities. Margaret Hutchinson Rousseau entered this crisis with credentials that made her simultaneously perfect for the challenge and completely unexpected as its solution. Born in 1911 in South Dakota, she had earned her doctorate in chemical engineering from Yale University in 1937, one of the first women to receive that degree from an institution that had admitted female graduate students only reluctantly. Her dissertation research focused on industrial fermentation processes, specifically the optimization of bacterial cultures for producing organic acids. The work had given her expertise in exactly the kind of large-scale biological systems that penicillin production required. Rousseau joined Pfizer's Fermentation Development Division in 1941, hired specifically to address the penicillin scaling problem that was consuming the company's research budget without producing results. She arrived at Pfizer's Brooklyn facility to find a demoralized team of chemists and engineers who had spent months watching fermentation tanks produce yields that made commercial production impossible. The previous month's best result had extracted 17 units of penicillin per milliliter of culture broth, less than 1/10 the concentration needed for economically viable production. The conventional wisdom among pharmaceutical researchers held that penicillin's low yields in deep tank fermentation were inherent limitations of the mold's biology. Penicillium had evolved to grow as surface colonies on solid materials, not as suspended cultures in liquid media. Forcing it to grow in deep tanks was fundamentally unnatural, and the mold's poor performance reflected biological constraints that no amount of engineering could overcome. Industry experts recommended abandoning deep tank methods in favor of surface culture techniques, growing the mold in shallow trays that could be stacked in climate-controlled rooms. Rousseau examined this recommendation and rejected it immediately. Surface culture might produce better yields per unit volume, but it could never achieve the production scales that military demand required. Treating projected battlefield casualties from the planned invasion of Europe would need penicillin production measured in tons, not pounds. Only deep tank fermentation offered any possibility of reaching those volumes within the time constraints that Allied military planners had established. Her approach to solving the fermentation problem demonstrated both rigorous scientific methodology and willingness to challenge assumptions that other researchers had accepted as fixed constraints. Rousseau began by systematically varying every parameter in the fermentation process, testing different mold strains, culture medium formulations, temperature profiles, aeration methods, and agitation speeds. Each experiment was meticulously documented, creating datasets that revealed patterns invisible to researchers who had been making incremental adjustments based on intuition rather than systematic analysis. The breakthrough emerged from observations that contradicted established theory about penicillium metabolism. Conventional fermentation wisdom held that molds required gentle agitation to prevent damage to their delicate cell structures. Rousseau's experiments showed that penicillium actually produced more penicillin under vigorous agitation that would have destroyed more fragile organisms. The violent mixing broke up mold clumps, increasing surface area exposure to dissolved oxygen and dramatically improving antibiotic yields. Temperature control proved equally counterintuitive. Standard protocols maintained fermentation tanks at constant temperatures throughout the growth cycle, But Rousseau discovered that varying temperature in specific patterns during different fermentation phases could double penicillin production. The mold grew most rapidly at 72 degrees Fahrenheit during its initial multiplication phase, but produced maximum antibiotic concentrations at 66 degrees during its later metabolic stages. Maintaining these precise temperature profiles required instrumentation and control systems that didn't exist in conventional fermentation facilities. The corn steep liquor that served as the primary nutrient source presented its own challenges. This industrial byproduct of corn processing contained complex mixtures of amino acids, vitamins, and minerals that supported mold growth, but its composition varied significantly between batches and suppliers. Rousseau developed assay methods to analyze each shipment of corn steep liquor, then adjusted supplementary nutrients to maintain consistent culture medium composition regardless of raw material variations. The quality control procedures she established would become standard practice across the pharmaceutical industry, but in 1942 they represented innovations that most chemists considered unnecessary. Contamination control required engineering solutions that pushed the boundaries of industrial practice. Maintaining sterility in laboratory glassware was straightforward. Achieving it in 1,000-gallon steel tanks was exponentially more difficult. Rousseau designed steam sterilization protocols that raised tank temperatures to 248 degrees Fahrenheit under pressure, killing all contaminating organisms without damaging the tank's structural integrity. She developed air filtration systems that removed bacteria and mold spores from the compressed air used for culture aeration, using glass fiber filters that had never been employed in fermentation applications. By March 1943, Rousseau's modifications had increased penicillin yields from 17 units per milliliter to 140 units per milliliter, an improvement that transformed commercial production from theoretical possibility to practical reality. But even these dramatic gains fell short of the production volumes that military planners projected would be necessary for the planned Allied offensives in Europe and the Pacific. The War Production Board had established a target of producing enough penicillin to treat 100,000 patients by June 6, 1944, the date set for the invasion of Normandy. Meeting that goal would require further innovations that most pharmaceutical executives still considered impossible. The Terre Haute facility represented Pfizer's commitment to industrial-scale penicillin production, despite persistent skepticism from competitors who believed the company was wasting resources on an unproven technology. The converted ice cream factory offered advantages that purpose-built pharmaceutical facilities lacked. Its refrigeration equipment could be modified for precise temperature control. Its stainless steel tanks, originally designed for dairy products, met the sanitary standards that penicillin fermentation required. Most importantly, the facility was available immediately, eliminating the year-long construction delays that new building would have required. Rousseau spent April 1943 converting ice cream production equipment into the world's first industrial penicillin fermentation facility. The 14 fermentation tanks ranged from 7,000 to 10,000 gallons each, volumes that exceeded the largest penicillin production vessels previously attempted by a factor of 5. Each tank required its own temperature control system, aeration equipment, agitation motors, and sterile feed systems. The complexity of operating these tanks simultaneously, while maintaining the precise conditions that successful fermentation required, presented logistical challenges that had never been solved at this scale. The facility's first production run began on May 1, 1943, inoculating all 14 tanks with Penicillium cultures that Rousseau's team had optimized through months of strain selection work. The fermentation would require 14 days to complete, during which continuous monitoring would track dissolved oxygen levels, pH, temperature, glucose concentrations, and a dozen other parameters that influenced penicillin production. Any significant deviation from optimal conditions in any tank could destroy the entire batch, representing weeks of wasted effort and thousands of dollars in lost materials. The results exceeded even Rousseau's optimistic projections. On May 14th, when the first harvest began, average yields reached 175 units per milliliter, higher than any previous industrial production attempt, and approaching the theoretical maximum that penicillium's metabolism could achieve. The 14 tanks produced in 2 weeks more penicillin than all of America's laboratories had manufactured in the previous year. More critically, the production costs had dropped to levels that made commercial distribution economically viable for the first time. The pharmaceutical industry's response to Terre Haute's success revealed how completely skepticism had dominated previous assessments of penicillin's commercial viability. Merck and Squibb, who had invested millions in surface culture methods, immediately began converting facilities to deep tank fermentation using Rousseau's published protocols. Pfizer's willingness to share technical specifications with competitors, mandated by War Production Board directives that prioritized rapid production scaling over patent protection, accelerated the industry-wide transition to methods that months earlier had been dismissed as impractical. But scaling from one facility to dozens required solving problems that extended beyond fermentation chemistry into realms of supply chain management, quality control, and clinical distribution that had never been attempted for any pharmaceutical product. Penicillin's instability meant it degraded within days unless stored under refrigeration, creating logistical challenges for military deployment to combat zones where refrigeration was unavailable. The drug's potency varied significantly between production batches, requiring standardized assay methods that could ensure consistent dosing. Most critically, physicians had no established protocols for administering penicillin, leading to treatment failures when drugs were given in insufficient doses or discontinued too early to fully eliminate infections. Dr. Chester Kiefer assumed responsibility for coordinating penicillin distribution and clinical research in August 1942, appointed by the Committee on Medical Research to manage what had become a national priority program. His position carried authority to allocate every unit of penicillin produced in the United States, determining which patients received treatment and which physicians gained access to the experimental drug. The ethical weight of these decisions intensified as production increased but remained far short of demand from military hospitals treating wounded soldiers and civilian facilities, managing bacterial infections that penicillin could cure. Kiefer's allocation decisions prioritized clinical research that would establish treatment protocols for battlefield medicine. Military casualties presented bacterial infections under conditions that civilian hospitals rarely encountered. Combat wounds contaminated with soil bacteria caused gas gangrene, a rapidly fatal infection that destroyed tissue faster than surgeons could amputate. Battlefield conditions delayed treatment, allowing infections to progress beyond stages where conventional antiseptics remained effective. Most critically, wounded soldiers required treatments that could be administered by medics with minimal training, eliminating complex protocols that civilian specialists could employ in hospital settings. The clinical trials, conducted during late 1942 and early 1943, established penicillin's effectiveness against previously untreatable infections, while revealing limitations that would influence how the drug was deployed during major combat operations. Streptococcal infections, the primary cause of wound sepsis, responded dramatically to penicillin treatment, with mortality rates dropping from 40% to less than 5% when the drug was administered within 24 hours of infection onset. Staphylococcal pneumonia, which killed 30% of patients in military hospitals, became survivable with penicillin regimens that cleared infections within 72 hours. But gram-negative bacterial infections, including those caused by Pseudomonas and Escherichia coli, showed minimal response to penicillin treatment. The drug's mechanism of action, disrupting bacterial cell wall synthesis, proved ineffective against organisms with different cell wall structures. This limitation meant that penicillin could not replace surgical debridement and antiseptic wound care, but rather complemented existing treatments by preventing secondary infections that conventional methods could not control. Dosing protocols required extensive trial and error to establish effective regimens. Fleming's original patients had received penicillin in doses measured in thousands of units, but battlefield casualties required millions of units administered over several days to fully eliminate infections. The drug's rapid excretion through kidneys meant that blood concentrations dropped to ineffective levels within 4 hours of injection, requiring dosing every 3 to 4 hours to maintain therapeutic levels. Military medical planners calculated that treating a single soldier with penicillin for a severe infection required 48 injections over 1 week, presenting logistical challenges for field hospitals operating under combat conditions. Production scaling accelerated throughout late 1943 as additional pharmaceutical facilities adopted Rousseau's fermentation methods. By December, monthly penicillin production reached 21 billion units, enough to treat approximately 400 patients with severe infections. This represented a 100-fold increase from June's output, but remained far short of the War Production Board's target of producing sufficient penicillin for 100,000 patients by the Normandy invasion date. The bottleneck had shifted from fermentation technology to downstream processing. Extracting penicillin from fermentation broth required complex purification procedures that removed proteins, residual nutrients, and metabolic byproducts, while preserving the drug's fragile molecular structure. Early purification methods achieved yields of only 30%, meaning that 70% of the penicillin produced through fermentation was lost during extraction and purification. Rousseau's team worked through winter 1943, developing improved extraction techniques that increased purification yields to 65%, effectively doubling the penicillin available from existing fermentation capacity. The chemical instability that had plagued penicillin since Fleming's original discovery required innovations in formulation and storage that would influence pharmaceutical practice for decades. Penicillin degraded rapidly in aqueous solution, limiting its shelf life to days even under refrigeration. Rousseau collaborated with formulation chemists to develop freeze-dried preparations that remained stable for months at room temperature, then could be reconstituted with sterile water immediately before administration. The freeze-drying process, adapted from techniques developed for preserving blood plasma, required specialized equipment and added costs to production, but provided the only practical method for stockpiling penicillin for military deployment. If you find this story engaging, please take a moment to subscribe and enable notifications. It helps us continue producing in-depth content like this. By March 1944, 9 months before D-Day, penicillin production reached 200 billion units monthly, distributed among military hospitals, naval medical facilities, and selected civilian institutions participating in clinical research. The allocation system remained tightly controlled, with Kiefer personally reviewing applications for penicillin access and prioritizing cases that would generate data applicable to battlefield medicine. This centralized control created tensions with civilian physicians who watched patients die from infections that penicillin could cure, while limited supplies were reserved for military applications. The ethical complexity of wartime drug allocation intensified when penicillin proved effective against venereal diseases that were decimating military readiness. Gonorrhea and syphilis infected thousands of soldiers monthly, removing them from combat duty for weeks of treatment with conventional arsenical drugs that caused severe side effects and required prolonged hospitalization. Penicillin cleared gonorrhea infections within 24 hours and dramatically accelerated syphilis treatment, reducing hospital time from weeks to days. Military medical officers advocated for allocating significant penicillin supplies to treating venereal disease, arguing that returning infected soldiers to duty quickly provided greater military benefit than reserving drugs exclusively for combat casualties. Kiefer's decision to approve penicillin for venereal disease treatment sparked controversy among researchers who believed the drug should be reserved for life-threatening infections. The debate revealed fundamental questions about medical resource allocation during wartime. Should treatments be distributed based on medical need, prioritizing patients with the most severe conditions? Or should military necessity determine allocation, directing resources toward interventions that most effectively maintained combat strength regardless of individual patient circumstances? The resolution established precedents for medical ethics that would influence healthcare policy for generations. Penicillin would be allocated based on military effectiveness, treating both combat casualties and venereal disease patients, with remaining supplies distributed to civilian cases that contributed to clinical research. The The decision meant that some civilians died from treatable infections, while soldiers received penicillin for non-life-threatening conditions. A moral calculus that wartime necessity imposed on medical professionals trained to prioritize individual patient welfare above broader strategic considerations. Rousseau's work during early 1944 focused on increasing fermentation yields beyond the 175 units per milliliter that Terre Haute had achieved. Her team tested thousands of Penicillium strains collected from sources worldwide, seeking natural variants that produced higher antibiotic concentrations. The breakthrough came from an unexpected source when a laboratory technician brought in a moldy cantaloupe purchased at a Peoria, Illinois market. The Penicillium strain isolated from that cantaloupe, designated NRRL 1951, produced penicillin at concentrations exceeding 250 units per milliliter under Rousseau's optimized fermentation conditions. Further optimization of culture conditions pushed yields to 300 units per milliliter by April 1944, nearly doubling production capacity from existing fermentation facilities. The improved strain was distributed to all pharmaceutical companies participating in the penicillin program, enabling industry-wide production increases that finally approached the War Production Board's targets. By May, monthly production reached 500 billion units, sufficient to treat approximately 10,000 patients with severe infections or 50,000 patients with moderate bacterial conditions. The logistics of deploying penicillin for the Normandy invasion required innovations in military medical organization that transformed how battlefield casualties were treated. Traditional doctrine called for wounded soldiers to be evacuated to rear area hospitals, where specialists could provide definitive care. But penicillin's effectiveness depended critically on early administration, with treatment delays beyond 24 hours, significantly reducing survival rates for infected wounds. This meant that penicillin needed to reach frontline medical units, requiring supply chains that could deliver refrigerated medications to combat zones and training that enabled medics to administer injections under battlefield conditions. Colonel Edward Churchill, chief surgical consultant to the Mediterranean Theatre, developed protocols for forward deployment of penicillin during the Italian campaign in late 1943. His system positioned penicillin stocks at battalion aid stations, the first medical facilities that wounded soldiers reached after battlefield evacuation. Medics received training in intramuscular injection techniques and dosing schedules that could be administered without physician supervision. Refrigerated storage units, powered by portable generators, maintained drug stability in field conditions where ambient temperatures exceeded penicillin's degradation threshold. The results from Italian campaign medical reports demonstrated penicillin's impact on combat mortality with statistical clarity that influenced planning for Normandy. Wound infection mortality rates dropped from 15% to 3% in units with access to early penicillin treatment, compared to units receiving conventional antiseptic care alone. Gas gangrene, previously fatal in 70% of cases, became survivable when penicillin was administered within 12 hours of wounding. Perhaps most significantly, soldiers treated with early penicillin returned to duty faster, reducing the manpower losses that infection-related evacuations imposed on combat units. Planning for D-Day incorporated these lessons into medical logistics that would support the largest amphibious invasion in military history. Allied planners projected 100,000 casualties during the first month of Normandy operations, with bacterial infections expected to affect 40% of wounded soldiers if conventional treatment methods were employed. Stockpiling sufficient penicillin to treat projected cases required accumulating reserves that exceeded total production from the program's inception through early 1944. The pharmaceutical industry responded with production increases that demonstrated how completely industrial methods had transformed penicillin from laboratory curiosity to mass-produced commodity. By June 1, 1944, monthly production exceeded 650 billion units, allocated primarily to building stockpiles for the invasion. Pfizer's facilities in Brooklyn and Terre Haute operated continuously, with fermentation tanks inoculated on staggered schedules that produced harvests every 2 days. Quality control procedures that Rousseau had established ensured consistent potency across production batches, eliminating the variability that had plagued early manufacturing attempts. June 6, 1944 arrived with Allied forces carrying penicillin supplies unprecedented in military medical history. The first wave landing at Omaha Beach included medics equipped with penicillin doses sufficient to treat 50 soldiers each, with resupply systems designed to deliver additional stocks within 24 hours. Field hospitals, established inland from the beaches, received refrigerated storage units containing penicillin, reserves measured in millions of units, enough to treat severe infections for hundreds of patients simultaneously. The invasion's medical statistics revealed penicillin's impact, with numbers that would have seemed impossible 2 years earlier. During June 1944, Allied medical units in Normandy treated 12,000 wounded soldiers with bacterial infections. Mortality from these infections measured 2.8%, compared to historical rates exceeding 12% in previous amphibious operations conducted without penicillin availability. Gas gangrene cases, which had killed thousands during World War I's trench warfare, numbered fewer than 200, with mortality rates below 5% when penicillin treatment was promptly administered. Rousseau received these reports at Pfizer's Brooklyn headquarters, while supervising production runs that now generated penicillin in quantities that would have supplied Fleming's entire research program for decades. The woman whose fermentation innovations had been dismissed as impractical now oversaw facilities producing 2 billion units of penicillin daily, enough to treat 400 patients with severe infections, or 2,000 patients with moderate bacterial conditions. The impossible had become routine through systematic application of chemical engineering principles to biological systems that conventional wisdom had declared unsuitable for industrial scaling. The broader impact of wartime penicillin production extended far beyond immediate military applications. Rousseau's fermentation methods established technological foundations for the modern pharmaceutical industry, demonstrating that biological manufacturing could achieve industrial scales previously reserved for chemical synthesis. Her quality control protocols became industry standards, ensuring consistent drug potency across production batches. The collaborative research model that had accelerated penicillin development, sharing technical innovations among competing companies under government coordination, would influence how future medical emergencies were addressed through public-private partnerships. By war's end, in August 1945, penicillin production exceeded 7 trillion units annually, sufficient to treat millions of patients worldwide. The drug that had barely existed outside research laboratories in 1940 had become so abundant that military medical units discarded partially used vials, a practice that would have been inconceivable just 3 years earlier. Civilian access to penicillin expanded rapidly during late 1945, as military demand decreased and pharmaceutical companies redirected production to domestic markets. Rousseau's technical contributions received formal recognition through awards and honors that acknowledged her central role in transforming penicillin from experimental treatment to mass-produced therapeutic. But the deeper significance of her work lay in demonstrating that systematic engineering approaches could solve problems that biological scientists had declared unsolvable. The fermentation methods she developed would be adapted for producing vitamins, amino acids, vaccines, and eventually the sophisticated biological drugs that would define 21st-century medicine. The human cost of delayed penicillin availability haunted medical professionals who had witnessed patients die from infections during the years when production methods remained inadequate. Statistical analyses conducted after the war estimated that penicillin's availability during 1944 and 45 prevented approximately 150,000 deaths among Allied military personnel, based on infection mortality rates from previous conflicts compared to World War II outcomes. These lives were saved not through medical breakthroughs in understanding bacterial infections, but through industrial innovations that made existing treatments available at scales matching military need. The counterfactual question of how many additional lives might have been saved if industrial production methods had been developed earlier remained unanswerable, but profoundly troubling. Fleming's 1928 discovery had revealed penicillin's antibacterial properties, but 15 years elapsed before Florey's Oxford team demonstrated clinical effectiveness, and another 3 years passed before Rousseau's fermentation methods enabled mass production. The scientific knowledge required for penicillin therapy had existed since the early 1930s, but translating that knowledge into practical treatments required industrial capabilities that pharmaceutical companies had been reluctant to develop without guaranteed markets. Rousseau's post-war career focused on applying fermentation technology to producing other medically important compounds. Her methods for cultivating penicillium in deep tanks were adapted for Streptomyces bacteria that produced streptomycin, the first effective treatment for tuberculosis. The temperature control systems she designed for penicillin production enabled commercial manufacture of vitamins through bacterial fermentation. Most significantly, her quality control protocols established regulatory standards that ensured pharmaceutical products met consistent purity and potency specifications, addressing safety concerns that had plagued the industry since its inception. The ice cream factory in Terre Haute continued penicillin production through 1952, when Pfizer consolidated manufacturing at newer facilities designed specifically for pharmaceutical applications. The building's conversion from food production to drug manufacturing had been intended as temporary expedient during wartime emergency, but the success of that improvisation demonstrated that effective solutions often emerged from circumstances that forced abandoning conventional tub approaches. The technical details of Rousseau's fermentation innovations, published in scientific journals during the late 1940s, revealed how completely her methods had departed from established biological research practices. Where microbiologists had treated fermentation as fundamentally biological process, requiring gentle conditions that mimicked natural growth environments, Rousseau had approached it as chemical engineering problem requiring optimization of mass transfer, heat exchange, and reaction kinetics. Her vigorous agitation methods, precise temperature cycling, and systematic quality control reflected industrial rather than biological thinking, applying principles from petroleum refining and chemical manufacturing to systems that most researchers still considered too complex for engineering analysis. Thank you for watching. For more detailed historical breakdowns, check out the other videos on your screen now, and don't forget to subscribe.